Hello and welcome back. After learning about glycolysis, a link reaction, and the Krebs cycle, we're now ready to go into the final part of aerobic respiration. Let's learn about oxidative phosphorylation, which is going to involve the electron transport chain and also chemiosmosis. As usual, before we go into the content, let's have a look at the learning outcomes. I hope that by the end of this lesson, you would be able to illustrate to explain the electron transport chain, and that involves all these things that we have on the screen here, and also explain what is chemiosmosis, and explain the complete oxidation of one molecule of glucose in active cells to produce 38 ATP. How does it get to 38? We'll have a look at that later. So let's have a look and learn at what happens inside the powerhouse of the cell. Now you remember what oxidative phosphorylation is. I've talked about it since 5.1. It's one of the ways that we are going to make ATP in the cell. So this thing, oxidative phosphorylation, it will involve electron transport chain and chemiosmosis, and it occurs in the inner membrane of the mitochondria, also called the crista. So if this is the structure of the mitochondria, the blue part is where we had the Krebs cycle, where oxidative phosphorylation will occur is here, on the inner membrane of the mitochondria. Now let's have a see what is an electron transport chain. An electron transport chain consists of a series of electron carriers embedded in the inner membrane of the mitochondrion or crista. So dalam inner membrane mitochondria, ada banyak electron carrier dan bila dorang pindah-pindahkan elektron, tenaga akan terhasil. So they will carry electron from the NADH and FADH2 that we had during the previous reactions to synthesize ATP. And there are several components in this electron transport chain. So saya cakap ada electron carrier dalam ETC. Siapakah electron carrier tersebut? Okay, so we have these main components. The main components, we start with complex 1, also called NADH dehydrogenase. So complex 1, this thing, it will accept electrons from NADH molecules, the tukang terima electron. These NADH were produced during glycolysis, link reaction, and Krebs cycle. NADH will transfer electrons to complex 1, NADH dehydrogenase. NADH will be oxidized and then they will become NAD+. And then NADH dehydrogenase will transfer electrons to ubiquinone, which is CoQ. The second component of the ETC is also called complex 2. Okay, ini adalah benda kedua. Complex 2 nama penuhnya adalah succinate dehydrogenase. So what does complex 2 or succinate dehydrogenase do? It will accept electrons from FADH2 molecules. Okay, so 2, FADH2. FADH2 were produced during Krebs cycle, and when it gets to complex 2, it will be oxidized and form FAD. And then succinate dehydrogenase will also transfer electrons to ubiquinone, also known as CoQ. Now let's learn about ubiquinone. This is the next guy that is involved in ETC. Ubiquinone, or coenzyme Q, nama singkatan CoQ, is a mobile electron carrier. The function of ubiquinone is to accept electrons from complex 1 and then transfer it to complex 3. Ataupun dia boleh terima elektron daripada complex 2 and then also transfer to complex number 3. And that brings us to the fourth character in the story, which is complex 3, Cytochrome C reductase. Okay, watak keempat ini, complex 3, fungsi dia adalah terima elektron daripada ubiquinone. So it's going to accept the electron from ubiquinone, and then it's going to transfer that electron to the next person, which is cytochrome C. Cytochrome C, watak nomor 5, is a mobile electron carrier. It will accept electron from complex 3 and then transfer it to complex 4. So ini watak ke-6. complex 4 is cytochrome C oxidase. It will accept electron from cytochrome C and then transfer that electron to oxygen, which is the last guy. So this character number 7 is oxygen molecule. This is the final and the last electron acceptor in the electron transport chain. So 2 hydrogen ions, H+, from mitochondrial matrix will combine with 2 electrons and half of an oxygen molecule to form water. And put that into a chemical equation, it will become 2H plus 2 electrons plus half or 2 becomes 1 molecule water. Okay? So, macam panjang di sini, dan kita akan jumpa watak-watak ini lagi sambil kita belajar pasal prosesnya. So, just now we were just getting to know the components that are involved. Now we will see how they work with each other. So, ini adalah diagram yang saya akan gunakan untuk explain electron transport chain and chemiosmosis. Kamu boleh cuba lukis sendiri. Dalam worksheet aerobic respiration pun ada diagram ini juga. Alright, so let's have a look at it step by step. Now let's look at how it starts. The NADH and the FADH2 that were created in the previous processes are going to be brought to the inner membrane of the mitochondria. So, dia akan dibawa di sini. Bila sampai di sini, NADH will transfer electrons to NADH dehydrogenase. Okay? So, get that electron, transfer it to complex 1, NADH dehydrogenase. After it transfers the electron, that means NADH is oxidized to NAD+. What happens on the next complex, complex 2? FADH2 will transfer electrons to complex 2, which is succinate dehydrogenase. After it gives away its electron, FADH2 will be oxidized to FAD. Electrons from NADH dehydrogenase, which is complex 1, or succinate dehydrogenase, which is complex 2, these electrons will be transferred to ubiquinone, CoQ, Cytochrome C reductase, Cytochrome C, and then Cytochrome C oxidase. So, kamu boleh nampak di sini, anak panah dia mula dengan complex 1, complex 2. Ini, mobile electron carrier yang pertama, ini adalah ubiquinone. Kamu nampak sini, yang ada nombor 3, ini adalah Cytochrome C reductase. Mobile electron carrier yang kedua di sini, atas sikit ini adalah Cytochrome C. Dan yang ada nombor 4 di sini, complex 4, this is Cytochrome C oxidase. So what this sentence is saying here, electron dari sini, kena passing. That is what is being described here. And then, when it reaches the last guy, which is Cytochrome C oxidase, ataupun digelar juga sebagai complex 4, Cytochrome C oxidase will transfer electrons to oxygen molecule, which is the final electron acceptor. So, dia dapat elektron daripada orang lain. Dia akan ambil elektron itu, masukkan ke dalam oxygen molecule. Pandai-pandailah oxygen molecule itu akan balance dengan benda di sekitarnya sampai dia jadi water molecule. So, in case you are ever asked what is the final electron acceptor in ETC, final electron acceptor in ETC is oxygen molecule. Dia adalah orang terakhir yang terima elektron. And then this oxygen will combine with hydrogen ion to form water, as I've said before. Now, why is it important for them to pass those electrons along? Because as the electron moves along the electron transport chain, energy will be released from the redox reactions. It's a little bit hard to imagine, but just think of it this way. When molecules are being broken or they're being passed around or being rearranged, sometimes energy will be released. Okay? Bila ada redox reaction, ada benda dipecahkan ka, digabungkan ka, diputuskan ka, akan ada tenaga dilepaskan. So, bila elektron kena passing, elektron dan energy akan dihasilkan. Oh, wait. More accurately, as the electron is being passed, energy will be released from the redox reactions. So energy here, energy here, energy here. This energy will be used by the protein complexes to pump protons or hydrogen ions from the mitochondrial matrix into the intermembrane space, causing the accumulation of high concentration of protons or hydrogen ions in the intermembrane space. So apa yang setiap kompleks ini buat dengan tenaga daripada elektron tadi? Dia akan gunakan tenaga tersebut untuk bawa hydrogen ion ataupun proton, okay? They're going to bring all this proton to this side of the membrane. So dia akan punggahkan supaya di sini ada high concentration of proton. So now there is high concentration of protons in the intermembrane space, and this creates a proton gradient. Sini high concentration, di bawah low concentration. So ada sudah proton gradient. Juga digelar sebagai proton motif force. So this creates a proton gradient across the inner mitochondrial membrane, and the inner mitochondrial membrane is impermeable to protons. Membrane ini, this inner mitochondrial membrane, proton tidak boleh suka hati limpas. So, kalau dia tidak boleh suka hati limpas, dia perlu cari protein yang membenarkan dia untuk limpas. So, this proton gradient will cause protons from the intermembrane space to diffuse or flow back into the mitochondrial matrix through ATP synthase, which is down its concentration gradient. So, pada membrane, inner mitochondrial membrane itu akan ada protein ini. Protein ini adalah ATP synthase. ATP synthase ini seakan-akan turbin dalam empangan. Okay, it's like a turbine in a dam. It will allow those protons to pass through. ATP synthase akan benarkan proton untuk limpas, dan bila proton itu melalui ATP synthase, dia akan turun mengikut concentration gradient dia. Dan dia akan pusingkan sesuatu di dalam ATP synthase. You can learn more about the structure on your own, but just long story short, when it goes down, it spins something. When it spins something, this protein has enough energy to do phosphorylation. So it's going to take ADP, a phosphate group, and it's going to generate ATP. Kamu tidak perlu explain panjang lebar lah. Kamu boleh explain dengan satu ayat ini sahaja. Tapi secara general, what happens? Proton limpas, tenaga terhasil, ATP terhasil. So this part on the left side is the electron transport chain. And this part in the green box for this slide is called the chemiosmosis part of oxidative phosphorylation. Now, you can try to draw this on your own. I know there's a diagram for it in the aerobic worksheet, but you should be able to remember it much better if you try drawing the diagram on your own. Now, in the next two slides, you will have this long chunk of text. This is the same thing that I was explaining before. It's just laid out sentence by sentence in case you ever need to do an essay. So, ini adalah benda yang sama yang saya explain tadi, cuma dia disenaraikan macam ini supaya mudah untuk kamu baca sekiranya kamu mau gunakan untuk jawab essay. Okay, I'm not going to read through all of it. So, yeah. Okay, now we're going to talk about the energy that is produced from one molecule of glucose in an active cell. So, macam mana kita tahu satu molekul glucose boleh hasilkan berapa molekul ATP? Kita akan kira dengan cara ini. So, we're going to look at each of the reactions that we've had and then see the products of the reaction. And then, based on the molecules produced, we are going to think of what happens during oxidative phosphorylation and calculate the number of ATP. So during glycolysis, we are going to produce 2 molecules of NADH and 2 molecules of ATP. Yang ATP terus bawa pergi sana lah, pergi total number of ATP produced, memang terus 2. Tapi kalau untuk 2 NADH, dia akan melalui oxidative phosphorylation. So the way we calculate it is like this. And it could produce 6 or 5 molecules of ATP. So kadang-kadang 5, kadang-kadang 6. Bergantung kepada kondisi kebetulan apa yang berlaku dalam mitochondria. Whereas from link reaction, we don't produce any ATP. We produce 2 molecules of NADH. That NADH, when it goes through oxidative phosphorylation, it could produce 6 molecules ATP or it could produce 5 molecules ATP. In Krebs cycle will produce 6 molecules of NADH, which would become 18 molecules of ATP maximum or 15 molecules of ATP minimum. It will also produce 2 molecules of FADH2. 2 molecules of FADH2 could produce maximum 4 molecules of ATP, minimum 3 molecules of ATP. And also, Krebs cycle produces 2 molecules ATP. Ito terus bawa pergi sebelah kanan. So, these, tambah. Daripada 1 molekul glukos, kita kemungkinan boleh dapat paling sedikit 32 molekul ATP, paling banyak 38 molekul ATP. So if anybody ever asks you how much ATP can you produce from 1 molecule of glucose, you can explain to them in this way and tell them maximum is 38 molecules of ATP. Kamu akan nampak nota kami kebanyakannya akan guna 38 molekul ATP. Dan the reason we do this is because we want to assume the maximum number of ATP produced from 1 molecule of glucose. Kalau kamu tidak ingat, ataupun macam kamu serabut berapa banyak molekul actually, just remember 38. 32 to 38 molecules of ATP. But usually, we just answer 38. So I hope that part is clear. Just a very quick summary of what we've gone through so far. In this video, we've already gone through the ETC and also chemiosmosis, which makes up oxidative phosphorylation in aerobic respiration. Dan lepas tu kita juga kira berapa molecules ATP boleh dihasilkan daripada aerobic respiration. And that's pretty much it for aerobic respiration. That's kind of all I need to tell you. You can, of course, keep going back to this diagram, have a look at your own notes, have a look at the worksheets that we've done. And what should you do now? I hope that you would complete the rest of the Chapter 5 aerobic worksheet. And you should also complete the tutorial questions for Chapter 5, especially in Part B. And I asked you to do a lot of drawings for this subtopic. Please submit your drawings for glycolysis, Krebs cycle, and oxidative phosphorylation through the submission slots in the Google Classroom. That's all for me for this section. Join me again in the next video for 5.2, where we learn about respiration that does not need oxygen. Thank you very much for your time and your attention, and I will see you again soon. Bye!